Good afternoon, good morning, everybody, wherever you are in this world watching. So thanks for joining in. First of all, I would like to welcome Sasha Klibanov as a co-chair of the session and all the speakers that are here on the panel live to discuss some exciting questions. And I hope for more questions coming in from the audience while we discuss and talk. So I think when I went through the talks, there was really exciting stuff going on. I'm really very impressed with the work that was submitted and that we had a chance to see here. And so let me kick off with some questions, and we will go by the order of the speakers. So I would like to ask the first questions to Avinoam on these genetically encoded cavitation. So thanks a lot for this interesting work. And if I understood correctly, these are pretty much proteins vesicles that have a dimension, I put it down roughly on the order of 500 nanometers length times 100 nanometers diameter filled with gas. So I was wondering, what is the circulation time actually of these proteins in the blood circulation? Especially because they are not of human or marine nature. Are there any immune effects that, for example, lead to rapid excretion?
Yeah, so thank you very much and good morning. With the original structure, gas vesicles circulate for just a couple of minutes before they are collected by the immune system. But in the last couple of months, years, we're able to coat them in different coatings that enable to use them up to, I think we got currently 30% of the maximum signal from gas vesicles in the bloodstream after 2 hours. So you can really extend the circulation. They are stable. The structure is stable. So it's all about evading the immune system, and you can do it quite successfully.
Okay, thank you. And I understood from your talk, these are RGD targeted, right?
Yeah, so we have two ways of using them, either as targeted, but I think that the more interesting way of using them is by expressing them. So the protein structures, and you can genetically encode them in bacteria and more recently in mammalian cells. So wherever these cells go, so does your gas vesicles go. So if you have something that is homing to tumor, for example, like an immune cell or a tumor homing bacteria, it will go into the tumor. And that's what kind of separates it from synthetic converse agents.
Well, I can see that for tumor homing bacterials, but for any tumor homing immune cells, you would first need to extract your immune cells, then genetically modify them and re-inject. Is that correct?
Yeah, so that's what people are doing now with, for example, CAR T cells. So we think that a similar pipeline could work. If you add your CAR T to your immune T cell, you could add also the gene responsible for producing gas vesicles.
And then the advantage would be, for example, to look at immune response, for example, of tumors, or what would be the application?
Look at immune response and have the ability to act on it, for example, creating cavitation or other bioeffects.
In the chat room, I see also the question if there are any chance of extravasation of those particles.
So we have different types of gas vesicle these days. I think that the tiniest ones can be extravasated. We're talking about things that are smaller than 100 nanometer.
Actually, I think that question was addressed at Dr. Coviello.
Was it? Oh, yeah. Sorry. We will come back to that question, but it was also here a good question. Thank you anyways. So, Sasha, I would like to give the word to you. Any more questions?
Maybe we should move forward. I think time-wise, we should, because we have many speakers for whom we have to, and we have limited time, really, right?
Yes.
So, so I guess the second question is for Dr. Coviello, and the question was at 450 nanometers, do you expect any extravasation from the blood vessels, or this is purely a vascularly targeted therapy? And if I could add to that my own question, like those intriguing particles, first of all, how do you make them? And then second question is, whether this relatively large thing, there is a spleen filter. So in my experience, if you have something larger than a quarter micron, even if heavily pegylated, it will go to spleen. What do you see in that respect?
So, okay, so I'll answer the first one first, and then I'll come back. We do see extravasation, so we have pretty convincing distribution studies where we see that not only in the case of no ultrasound that there is some extravasation, and depending on what cell type you're talking about, your leaky vasculature, your gaps in the endothelium can be have quite variable sizes. So we do see that, but more so we're expecting that we're not looking for EPR effect. We are actually expecting active pumping of the particles and whatever drug out of the vasculature, and we do see that. Your second question, is there a spleen filter? Well, we know some of them go to the spleen, but we do see them in our target tissue in the liver as well. So we see, quite extended lengths of time that we can cause cavitation for. So for what we consider to be a standard human dose, we can sustain cavitation reliably for upwards of an hour.
Upward of an hour. That's nice. Yeah. So what is the dose, human dose that you expect? Because this is polystyrene, if I remember correctly, right? No,
it isn't polystyrene. I mean, it's mainly PMA.
PMA. Okay, so it's biodegradable. Good.
So, sorry. Specifically, I'm sorry. What was your question? How they are made, or is it because that was one of the questions, I believe.
Yeah,
it's mainly a seed polymerization process, and you can read about it in our published literature, which is on our website.
So I guess next question from the chat is to Dr. Castillo. What is the benefit of performing 2 hyperthermia treatments rather than one longer one?
Okay, I would rephrase this question actually a little bit, because the reason we performed 2 hyperthermia treatments, the main reason was we just wanted to know at which time point during the whole infusion and the whole time of the experiment, at which time point does it make more sense to start a hyperthermia treatment. And that's why, in order to economize in animals, and so for animal animal protection reasons, and not to use too many animals, we performed therefore 2 hyperthermia treatments on the same animal, so that we could compare. Okay, does it make more sense to do it at minute 10 or to do it a little bit later? And as you can see in the results, of course, we get much higher concentration of the drugs. Especially for doxorubicin, the earlier we start. Of course, it would be in theory possible to do a longer hypothermia treatment of 1 hour, but then you have the problem that at the later time points, the temperature maps become more and more unreliable. So that wouldn't actually be very advisable.
Okay, thank you. And another question is, maybe you could say a few words about those liposomes, this DPPG2, that's something new and exciting. Maybe you could say a few words about circulation time, thermal response, etc.
Yeah, so the DPPG2 is a novel lipid that was developed by some cooperation partners in Munich, and it's being right now the clinical studies for this lipid and for liposomes containing this lipid are being pursued by a partner company called Thermosome, and we work with them. And so this lipid has a melting point, which is very good for what we want, because the melting point is around 42 degrees something. So it's just perfect for hypothermia treatment. And the liposome formulations were made together with DPPC and DSPC in a mixture, so that the melting temperature of the liposomes themselves was also within this range. And the liposomes are very stable at normal body temperature at 37 degrees. We have very little leakage. For cisplatin, we have like very, very little leakage. For doxorubicin, a little bit more. So at 42 degrees in both cases, the in vitro experiments that we didn't show in this presentation showed that we have almost immediate release in both drugs. And in vivo, of course, you have other conditions that made the release maybe not perfect in the case of cisplatin, for example, but in the case of doxorubicin, you see that we get really high concentrations.
How about circulation time?
Circulation time, as you can see in the pharmacokinetics graph that I have in my presentation, we get up to minute 180, we still have around 15 to 20% of the liposomes still in the bloodstream. For the liposomes themselves, the circulation time doesn't change very much. That's something that we have seen in rat experiments. It doesn't change very much depending on if you use the same 3 lipids for the liposomes, it doesn't matter that much. The circulation time remains more or less the same.
Okay. Thanks. So Holger, maybe you want to have questions and proceed further within next.
Well, I think Juan already covered all the questions I had on my list. So I think also in view of time, I would like to go to the next talk. Cheon Kim. I think this also is in the line of Juan's talk with temperatures induced drug delivery in this time in the brain. And here I have a couple of questions, especially about your heating. I think you showed the maps of PRFS measurements, MR measurements in the brain. And I wonder if you could comment on any heating near the skull.
So certainly that aspect is to we did the demo assessment as well with the H&E staining, and although we were when we do the heating experiment and we see the temperature mapping, we will see the little bit of increased temperature over the skull. However, that is why we started our study with the optimizing frequency to actually increase the intensity ratio between the inside the brain that we want to target over the skull area. And we used a optimized frequency to have the maximum effect. And we were able to kind of minimize the skull heating with the statistical mice model we used.
So could you briefly comment on the best frequency you found? Because you have two directions. If you go to lower frequencies, you will have less absorption by the bone. So right. That reduces the heating of the skull, I would say. But your focus spot gets larger. So if you're in a mouse, probably the skull and the bone is still in your focus area. If you go to higher frequencies, you get a much better focus. But on the other hand, higher absorption in the skull. So what came out the best for you?
For the specific B6 albino model that we used, we found that around 1.6 megahertz to be the optimal frequency that can compensate the local zone versus the skull heating.
And last question, could you comment on the clinical translation on that one? Simply, we have these brain systems that are used for ablation, for example, within the brain. But I wonder, ablation is done, let's say, within a few seconds, but hyperthermia lives from prolonged heating. So I would say at least 10 to 15 minutes for good drug delivery. So is that feasible? Can you do hyperthermia in the brain using those systems without heating too much the bone?
Certainly that is feasible. I think that compared to the mice model, so the mice skull is relatively much thinner than the human skull. So we were able to use just a single element transistor with the optimal frequency. However, if you go to the clinical level, the skull for the human is much thicker, and you have to incorporate all those skull aberration corrections. And if you are able to do that with the multi-elements possibly, I think it could be certainly feasible to do the hyperthermia treatment with the clinical level as well.
Have you run any simulations on that one? Because for me, it sounds a little bit counterintuitive because you're looking at 15 minutes versus, I mean, even in the ablation case, you have a cooling cushion on the skull to keep the skin temperature low. So have you done any simulations on that one?
With the skull human geometry, I have not, but I think that perspective or that part is certainly interesting to look into. So for the future work.
Okay, thanks.
Possibly.
And let's see, is there, I think in view of time, I would like to move to the next talk. Sasha, would you like to take that one?
So the question for Dr. Mohammadabadi is, how does IFP relate to IFV, and can you attribute the enhanced delivery to a pressure-based convective effect or diffusion due to a lower hydraulic pressure? And please be succinct because we have very little time left.
Okay, thank you. It's a very good question. You know, it's a long story, and we know that different parameters are involved in the increased interstitial fluid pressure in tumor. However, in our technique, previous studies they show that, you have elevated tumors from periphery toward the center of the tumors, and it's something that other researchers have shown. But so there are a lot of things about that. But in our study, we showed that using pulse-focused ultrasound, that it's pure mechanical, and since we are using low duty cycle, so we hypothesize that it's the mechanical effect. You can expand the extracellular matrix and also disrupt the collagen network. All these parameters, factors together, increase the hydraulic conductivity and also improves the nanoparticle and the drug delivery toward the center of the tumor. And we showed that, for example, in our study, we could get up to 57% improved nanoparticle delivery to the center of the tumors in head and neck tumors.
Thank you. So next question is for Cameron Smith. Do you expect this linear relationship to apply to other types of therapeutic agents like small molecules, drugs, viruses?
It's an interesting question. I can only talk about the parameter range I've explored, and there's certainly a lot more work that could be done on this. But I would have expected that you would continue to get, at least within this similar cavitation regime range, to get at least a linear relationship between the delivery and the cavitation dose based on the studies that I've done. But just because you get a linear relationship to the delivery doesn't necessarily mean a linear relationship to effect, such as, for example, with viruses, a linear increase in delivery could easily give you an exponential change in result.
Thank you. So Holger, would you like to pick up the stream?
Yeah, so the next one would be for Claire Wonker, also on temperature-induced drug delivery using rhabdomyosarcoma model and MR-HIFO. Here I have a very simple question. Your volume that you extract, the volume that you heat is much smaller than the volume of the tumor. So first question is, do you excise the entire tumor, and the values that you report are tumor average values?
Yeah, so we excised the entire tumor, and then we reported the average value of the drug within that entire tumor volume or mass.
That also means that if your tumor size is varying compared to your heated volume, you will get a lot of deviations from that one.
Yeah, so we actually accounted for that when we did our analysis, and there was no statistical difference between our groups in terms of tumor size.
And is there also no statistical difference of your heated volume?
No, so we use
depends usually on the orientation of your tumor sometimes. Huh?
Yes, it does. We kept a standard tumor or a standard heating region of interest for our tumors, so we didn't change that between the tumors to account for that.
Okay. And you never showed in your presentation the PRFS returning to baseline temperatures, 37 degrees.
Yeah, so we had a rapid return to baseline temperature after we stopped heating, but I didn't include it in that graph.
Okay, thank you. Okay, Sasha, would you like to continue?
Well, so what this is a question for Dijuan. So we're running very low in terms of time. So Dijuan, what kind of microbubbles did you use in your study?
So this is a simple question. So the microbubble I used in my study is a homemade microbubble. So it is around 4 to 5 micrometer. just we isolate the size of the bubbles.
Yeah, and the question to Joanna is, could these, well, we have 10 seconds left, so I guess we have to hold a smile and wait for Q. So I'm sorry we didn't have we slightly ran out of time. My apologies.
No problem.
For being a little slow. Yeah, but I guess we can communicate through the chat function later. There are questions there that we could take advantage of. And thanks very much for the great session, and Holger, great work at keeping everybody in check. And
maybe I didn't do such a great work because we're running out of time.
Yeah. So wonderful session, lots of things to where we can contribute, and we'll eventually help some patients.